Okay. Hello, everyone. My name is Jing Li. I'm now currently doing postdoc research in the lab of Professor Kath-Schorr in University of Köln in the chemistry department. So today I'm going to give a talk about innovative RNA research and therapy. So today's talk will include three parts, and the first part will be on natural base pair TPP3 and M for RNA modification. And the second part will be genetic alpha expansion of DNA with this pair of unnatural bases. So the work about those two parts have been already nicely done in our lab and already been published. And the third part, I'm going to present some preliminary results of a project that we are currently still working on. It's about the novel RNA modifications that we developed for therapeutic use. So let's get started. As we all know that DNA in the living organism on this earth basically includes 4 genetic aspects like ATCP, like they pair two natural base pairs. That to expand the genetic aspects, like scientists developed natural base pairs like TPP3 and NM, like for research purpose or further medical research. And when you incorporate those two unnatural bases into DNA, they are compatible with DNA polymerase for amplification. And those two unnatural base pairs are also compatible with the transcription machinery, which means that they can be site-specifically incorporated into RNA. And additionally, you can also add this reactive handle like to the unnatural base to further expand the application of RNA. So basically, for the unnatural base pairs, when they inverted into a nucleotide building blocks, they can be site-specifically incorporated into RNA by enzymatic processing under native conditions. And this is actually independent of the length of RNA that we want to generate. That the additional small reactive handle that can be attached on the unnatural base pair could even bring the bio or orthogonal chemistry reaction into the system, which allows the visualization of RNA in vivo by the fluorescent conjugated reporter groups. So first, I'm going to introduce how we specifically introduce cyclopropanol conjugated TPP3 into small size of RNA. So basically, first we have to prepare the DNA template for that. And for this purpose, we generate DNA template by solid phase synthesis with sites specifically modifying NM, which is a unnatural base. And then when the DNA is done, we use it as a template for in vitro transcription. And then we get the TPP3 site-specifically labeled RNA. And further with the INDGA click chemistry reaction, we could in vivo visualize the RNA. And this could be done by the reaction with the tetracycline conjugated fluorescence. So this application have been already shown in the work published in 2016 that we site-specifically incorporated the TPP3 in the tRNA, and which you could see from the fluorescence gel scan that the transcript is very pure. And for mid-range RNA labeling by CPTP3, we have to use the two-step assembly PCR, which includes also the overlapping DNA template that incorporates NM, which is the unnatural base. And once the DNA template is generated by PCR, we similarly do the in vitro transcription, and by using the IUTTA quick reaction, we can visualize RNA in vivo. So this has been also shown in publications that we successfully site-specifically incorporate 2 molecules of the TPP3 in the bacteria are well-defined GLMS, like shown here. And this is actually without interfere with the cleavage function at the site. As shown in the fluorescence gel scan, which you could see that in the presence of the binding protein GLC-N6C, that the modified ribozyme could self-cleave itself, to a efficiency like 70%. So then for long RNA, like incorporation of the CP-TP53, we basically use primers that generate the template from the plasmid. And of course, we have to use a reverse primer with site-specifically incorporated DNA. And this is also successfully shown in the publication that the sites have specifically incorporated this modification to the long non-coding RNA exists. And we also can visualize the RNA in vivo. So until now, I introduced the method that how to incorporate CP-TP53 in different lengths of RNA. And then it will be also interesting to apply it to mRNA, because mRNA has been developed into very powerful therapeutic, for example, against COVID-19. And now it's also under active research to further apply to against cancer or infectious disease and so on. So in our lab, that we also try to put this modification to mRNA, like shown here. We basically incorporate this modification of two molecules to the 3' UTR of the anterior mRNA. And post-transfection to HeLa cells, we could nicely see the signal of mRNA has been delivered into HeLa cells and the distribution of this mRNA. And to our splice, we also see that this modification increases the protein production of anterior fluorescent protein, which means that this modification might increase the translation efficiency of mRNA. And we also try to combine the natural-based modification with the natural-based modification, for example, uridine and 5-methyl-cytosine, and to see what happens. And here we could nicely also see the signals of mRNA, and also it increased the translation efficiency of the anterior mRNA. Moreover, that with the quantification of mRNA in HeLa cells post-transfection, like we quantify the mRNA with qPCR, like we could see in general, the modified mRNA has the lower transfection efficiency, but you could see at the 6-hour post-transfection lower amount in cell. And we also see that the modified mRNAs will have higher stability. And from the protein quantification, we see that the mRNA with combined modification shows actually high levels of the protein expression level, which means that the combined modification gives like lower transfection efficiency, higher RNA stability, and also higher translation efficiency. And this could be potentially used for therapeutic use for mRNA therapies. And then we switch to the XNA world. So basically, RNA modification are mostly based on the changes on the base, sugar moieties, or the phosphate backbone. And beyond this, it might be interesting to develop other modifications that out of this category. For example, PNA, that shows here, is a peptide nucleic acid, which totally replaces the sugar backbone, phosphate backbone with peptide light chain, which is a very revolutionary modification of RNA. And to achieve more or explore more modifications like this, that's tRNA caught our attention because it's highly because it's highly resistant to enzymatic hydrolysis, and it contains the resins instead of the ribose. And we try to combine this modification with our own natural base modification, like to generate or expand the XNA world at least the DNA genetic effects. So the former PhD student Hannah, she did nicely work. She tried to generate the natural nuclear-based triphosphate and wanted to incorporate it into DNA transcripts by using the DNA template, which has a complementary natural base. And this experiment called primer extension, and you could see the transcript afterwards by the fluorescent that is conjugated at the 5' of the DNA primer. And also the products could be confirmed by LC-MS afterwards. So first she tried to put TP3 on the DNA template, and then try to get the DNA transcripts with NAM. And that's shown on the gel that with the called RSGE polymerase that even or neither with or without mechanism, that she was not able to see the full length product. And this means that NAM is hard for the polymerase to extend the product. But for the LC-MS characterization, they found that actually the NAM triphosphate has been successfully incorporated into the transcript. So there is no extension, but there is incorporation of this on natural base to keep the name. And on the other way around, she tried to put NAM on the DNA template and try to get the transcripts with TP3. And this time it's lucky that by adding a mechanism, she could see the full length product, which means that TP3 could be successfully incorporated, and also the transcript could be extended. And then is the third part of our talk. So I would introduce the novel chemical RNA modifications that we developed for therapeutic use. So first we applied the modification to mRNA, and we expect higher translation efficiency and also higher stability of the mRNA. So basically, we generate the mRNA in vitro with modifications and then encapsulate them with lipopectin. And after cell transfection, we quantify the fluorescence protein product after 48 hours, or we analyze the mRNA stability after 6 hours, 24 hours, and 48 hours by qPCR. So as shown here, that basically with the modification X and also Y, we got more protein products, which indicate that improved translation efficiency of our RNA modification. And by mRNA quantification, we actually didn't see stability of the mRNA or modified mRNA, but instead we see that at 6 hours post-transfection, there is differential levels of mRNA amount in cells, which indicating different transfection efficiency for modification X of mRNA. And then we apply this modification to siRNA to see if it can efficiently knock down the target mRNA and also see if it's more stable. So it's a similar process as the mRNA. And it's shown here that in comparison with the negative control, if they didn't put any siRNA in the system, that the modified siRNA could nicely knock down the target mRNA. And for siRNA stability, we try to relate the siRNA amount at a later time point, like 8 hours after transfection, to an earlier time point, like 4 hours post transfection. We could show that with our modification, that the siRNA shows at least 2 times of stability compared to the wild type. So thank you for the attention. So this is our lab members and also our collaborators. So if you are interested in our research, feel free to contact us. Thank you.